Hi everyone, welcome to week 9. So week 9, what we're dealing with is microbiology, and I'm also going to focus on the throat culture collection and how to run a rapid strep test, partly because those are the labs that we're going to do this week, okay? So microbiology deals with chapter 8, and it's important to know what microbiology is, and that's really the study of microorganisms, which are tiny, unusual microscopic entities and that can carry on a living process. Things like bacteria, viruses, parasites, fungi. All of those are examples of microorganisms. When we talk about microorganisms, it's important to know that they really are all around us. They're in our food, in our water, in our soil, in the air, on our bodies, in our bodies. They are all over. Okay? When we talk about the ones that are in or on our body, it's important to know that those are normal. They're supposed to be there, and those are called normal flora. An example of that would be something like lactobacillus. Okay? So now that we understand microbiology and microorganisms, it's important that we know how to classify those microorganisms. So the first microorganism classification is a virus. And viruses are really the smallest entity form of a microorganism. They consist of RNA or DNA, which are what we find in our bodies, and they need a host cell to live on, and then they use that host cell's metabolic machinery in order to reproduce itself. So because of the fact that it needs that tissue cell or that host cell to live off of, they cannot be grown in a culture media. The nutrients that are found in a culture media really do not help a virus grow. Second classification will be a bacteria. Now bacteria are considered prokaryotic, which means that they are unicellular, but they do not have a true nucleus or a nuclear membrane. They do, however, consist of a cell membrane and a cell wall. Bacteria generally reproduce by what we call binary fission, which is an asexual reproduction in which the bacteria will split itself in half, or the cells in half, and in half, and in half, and in half, and continue to grow. That's why the bacteria grows very rapidly, and it grows very well on a culture media because of the nutrients that are found within the culture media. Helps the bacteria reproduce itself very quickly. Now when we look at bacteria after it's grown, we notice what are called colonies, which are just mass forms of bacteria grown together. Third classification of a microorganism is a fungus or a fungi, and these are considered eukaryotic. So what that means is they do actually possess a true nucleus and a true nuclear membrane. Unfortunately, fungi actually lack chlorophyll, so that stuff that's normally found in plants that help them reproduce, fungus don't have. So they actually like to grow in damp, dark places. They can grow on culture media with certain nutrients, and the most common forms you'll find will be things like mold or yeast. And the final classification would be something like a parasite. So parasites are really organisms that live on or in a host, and they get their nutrients from that host. Now, parasites can be single cell or multi-cell. An example of a single cell would be something like an amoeba, or if you've ever watched SpongeBob with your children, Plankton, he is an amoeba. He's a single-celled microorganism. Multi-celled could be things like roundworm, ringworm, or tapeworms. So those are our 4 classifications of microorganisms. But now that we know how to classify them, we have to know how to identify them. And we identify bacteria or microorganisms excuse me, by their growth pattern and their shape. So there are 3 different types. The first one is a coccus, and coccus is going to be round in shape, and it's generally going to be found in a single pair, a tetra, a chain, or a cluster. The most common coccus is going to be something like Staphylococcus or Streptococcus. Those are the ones that you'll hear most frequently. Second classification is going to be a bacillus, which is generally a rod shaped, and they are found in pairs, and it can combine with the cocci to become coccus bacillus, which gets very confusing to say, but just know that that's the combination of cocci and bacilli. When we talk about bacillus, the most common forms that you'll find this in are going to be UTIs, gastroenteritis, typhoid fever, whooping cough, gangrene, items like that. The final form and shape will be a spirillum, which is just what it sounds like. It's a spiral in shape, but it can also be curved around in a corkscrew or in a spring shape. And the most common form of this would be syphilis or cholera. We don't see cholera too much anymore, but you might still see syphilis, unfortunately, in this area. So it'll look like a spiral shape. Okay. All right. So now that we have discussed bacteria, the classifications, and the shapes of it, we need to discuss this week's lab, and that's going to be dealing with collecting a throat culture and running a rapid strep test. So when you collect a throat culture, this is actually the most common procedure that you will perform in microbiology within a physician's office laboratory. The reason is that you're probably going to be testing for strep throat, which happens very frequently in a physician's office. You will have the patient sit down in a chair, and you're actually going to stand above them, and you're going to have them tilt their head back, stick their tongue out, and say, ah. The reason is when you say, ah, it actually opens up your tonsils in the back of the throat so you can actually get at them. You're going to also use a sterile tongue depressor to hold the tongue down, because the tongue's natural reaction is to protect the throat when things come into it. So if you're not holding the tongue down when you go to swab the back of the throat, the tongue will come back, and you will actually get the tongue on the sample. It's very important that you do not touch the tongue or the teeth, because the tongue and teeth contain normal flora, and we don't want that on our sample, because then it will not have a true sample. Okay. When you're doing and collecting the throat sample, you want to do what we call a figure 8 pattern. So it's essentially an 8, a side 8 in the back of the throat. Okay. But not only do you want to do that figure 8, when you do it, you want to roll the specimen swab between your fingers. The reason is that it coats the whole swab, not just one part of it. And you're going to do that figure 8 around the patient's uvula in the back of the throat. Now, uvula is that piece that hangs down in the back of the throat for you. Okay, you'll see that a lot you'll see it on Thursday when we actually start performing these on one another. So now that we have our sample, we want to run what we call a rapid strep test. So strep tests look at strep species, and they are actually gram-positive cocci, and they're generally found in chains. Strep can be divided into groups from A to O, but this is according to how their antigenic properties show up. We will be dealing with group A, which is going to look for a microorganism called S. pyogenes. Okay? That's really what's used to detect strep throat. And when we're looking for strep throat, we want to look for symptoms such as, obviously, a sore throat. They're going to have a bright red pharynx in the back of the throat. The tonsils are going to have white patches on them. You're going to have your patient's going to have swollen glands, feeling very tired, or even having muscle aches. And it's very important that you tell your patients that strep is actually something that's very contagious, and it gets transferred by saliva or nasal secretions. So it's very important that if someone is diagnosed with strep throat, they do not share drinks with anyone else, because that can actually help spread the disease around. All right, so this week, that's kind of the overview that I have for you guys. I want to remind you that you have one discussion post for this week that will be due on Wednesday with your responses on Saturday at midnight. Also this week you will have your final quiz of the quarter, and we will do a review in class prior to that, so I do not fear anything but high grades on that. Also, it's week 9, so that means that your final diversity project and your final resume project are due. Please remember that your resume needs to be not only submitted to me in a Dropbox, but you also have to email Deb a copy. Reason being, she's getting ready to start sending you guys out for externship, and she needs to send out a resume in order for them to call you for an interview. So please make sure that you have your final draft resume submitted not only to me, but to Deb as well. And if you need her email address, I can give you that in class this week. All right, guys, I look forward to seeing you all on Thursday. Have a wonderful week. Bye-bye.